My name is Ed Levin. I'm a professor of psychiatry and behavioral sciences at Duke University Medical Center in Durham, North Carolina, in the United States. And it's my privilege and honor to address the Iranian Congress of Physiology and Pharmacology. And thank you so much for inviting me to speak to you here today. So today I'm going to speak to you about research in neurobehavioral toxicology and what it is and why we do it and what our findings are and why we think they're important. So just as an outline to preview the talk, I would like to talk to you a little bit about what is neurotoxicology and why it's important, the neurotoxicity of pesticides in particular and effects on sex differences, the recent developments in complementary models, including zebrafish in neurotoxicology, and the neurotoxicology of natural chemicals and also therapeutic treatments. So what is neurobehavioral toxicology? So it's a dysfunction caused by exposure to chemicals, including environmental pollutants or side effects of therapeutic drugs, including drugs of abuse, or even agents that aren't chemicals at all, such as radiation. So like any organ in the body, the brain can be affected by external influences and producing dysfunction. So neurobehavioral toxicology is a study of adverse effects of environmental chemicals and drugs on neurobehavioral function in a race to identify how these toxicants impair our sensory, motor, emotional, and cognitive function before we become too deluded to figure it out or even care. So just a little history of this. There's pollution you can see. This is what most people think about environmental pollution. It comes from smokestacks. This is a picture of Donora, Pennsylvania, about 100 years ago. This is where I grew up. My father was a physician in Donora. This was from before I was born. And there were still mills there when I was growing up, and it wasn't quite this bad, but it was pretty bad. I mean, it was in a valley and abandoned a river, and it was a natural bowl that would fill up with smoke with the when the environmental conditions were inverted and caused lots of respiratory and other sorts of impairments. And so this is what most people think of pollution you can see. But I just wanted to point out to you that there's also pollution you cannot see. This is a picture of Love Canal in New York, and you can't see the pollution, but you can see the results of the pollution. You can see the boarded up windows of the houses and this digging device here that is digging up the contaminated soil. And basically the area had to be abandoned because of the chemical dumping of the polychlorinated biphenyls and other persistent organic chemicals that are cancer-causing, also causing neurobehavioral impairments and other sorts of diseases. So there's pollution we can't see that does harm us, but we have to be aware of. And then there's strawberry fields. So like the Beatles song, Strawberry Fields Forever, yeah, forever. They, unfortunately, even though it's beautiful, this is in Watsonville, California, with the lush green strawberry fields and the beautiful, beautiful strawberries, very delicious looking in the lower right-hand corner. And why are they so flawless and beautiful? On the upper left-hand corner of the picture, you can see why. There's very heavy use of pesticides in these fields, including insecticides and other sort of pesticides to keep the berries, in this case, unblemished and beautiful. But as you can see in the center of the picture, there are the people who work there. And these are people picking the strawberries who are exposed to these chemicals and suffer the ill effects of this chemical exposure. And also the people who eat the strawberries. And, you know, it helps to wash things that you eat, but sometimes it's actually in the fruit and berries and vegetables as well. So granted, we need the pesticides to protect our food crops and our non-food crops, but we also need to do this intelligently and to do it with as low risk of use as possible. So our part of the process is to try to identify the adverse effects of this sort of chemical exposure in neurobehavioral systems, and to try to help with the decisions to lower the risks of health impairments. So it's not just environmental contaminants that can cause neurobehavioral toxicity. This isn't the main theme of the talk, but I'm going to talk about environmental contaminants. But I also want to make you cognizant of the problems that drugs can pose. And they can have persisting impairment for neurobehavioral function, both in terms of the adverse side effects of drugs that are given therapeutically, and with therapeutic drugs, it's always a balance between the risk of side effects and the benefits of the main effects. So we just need to properly gauge those. But also, there are the drugs of abuse, including nicotine in the case of tobacco, or opiates, or cannabinoids in the case of marijuana, stimulants such as amphetamine or cocaine, or other drugs like alcohol. These are particularly dangerous neurotoxicants because they not only cause toxicity, but the toxicity that they cause are to neurobehavioral systems that cause people to seek out more intoxication. So there's this vicious feedback loop where there's this ever-increasing toxicity that includes the person being intoxicated. But back to the environmental contaminants. So this is a graph of chemical production. This is a U.S. In the United States, but it's actually reflected worldwide in terms of synthetic or organic chemicals and the increase in production over the last 80 years or so. And you have this very parallel increase, in this case, of diabetes. And it's a very similar effect in terms of obesity, and that those two are probably related in terms of type 2 diabetes. And it's, you can say there's increasing obesity because of increased eating and decreased exercise. But there are also neural systems that are involved in regulation of appetite. So I'm not saying that it's completely involuntary. They're certainly a part of the system that involves refraining from overeating and eating correctly, but there are also systems in the brain that control and drive our appetite. So this is a part of the increase. And it's not just metabolic diseases such as obesity and diabetes, but also neurobehavioral impairments, in this case autism, that there's ever increasing rate of autism. And we see this over the last 50 to 80 years. And it's not just an increase in diagnosis, but also an increase in incidence. So as diagnosis has changed, when I was a child, the diagnosis was, quote, mental retardation. But that has been supplanted by other diagnoses that are more specific in terms of things like autism or attention deficit hyperactivity disorder, other sorts of developmental neural behavioral impairments. But the incidence of them has increased dramatically over recent decades. So how is this related to environmental contaminants? So just to give you an idea about the scope of the problem, 80,000 untested chemicals in our environment, industrial chemicals, and 2/3 of those are under-tested. And from the ones that have been tested, but it's estimated that 1/4 or 25% may be neurotoxic. So what we don't know about chemicals is a lot. So this graph shows that the great majority of the chemicals, we just have no clue as to their particular effects on the neurobehavioral function or even other biological functions. And there are very few that we've found that actually have characterized in terms of their toxicity. And so today I'm going to spend the initial part of the talk on organophosphate insecticides. So these are very widely used pesticides, and in particular, I wanted to start off with one called diazinon, which is organophosphate pesticide, very widely used, and this is the chemical structure. So what we found is the early developmental diazinon causes very long-term neurobehavioral toxicity. So exposure during early development from the early prenatal period through birth causes effects on neurobehavioral function that can last a lifetime. And we've done a lot of these studies in rat models. And here's an example of a cognitive test. This is novel object recognition. So this is just a diagram from the Bevins and Bashir paper showing a little cartoon diagram of a rat and in a chamber with two similar objects, and it has a period of time, typically about 10 minutes, to to look at the different objects and investigate them. They're naturally curious animals. And then after a delay, not 1 hour delay is typical what we use, they're put back into the chamber, but with one familiar object and one new object. And typically the rat goes to the new object. And so this is a good test of, do they remember where they what they've investigated already, and go to the new one. So we can see the effects of gestational exposure, prenatal exposure, early postnatal exposure to novel object recognition. So you can look at the familiar object versus the novel object. And the familiar object in the gray stipple bars showing with the zero exposure of the control group, there is a clear difference in terms of the investigation of the animal to the new object versus the old object. And the low dose, a similar pattern. But if you up it a little more, to 1 milligram per kilogram per day, you see that this preference is wiped out. So they really don't recognize the novel object at all. And so you have an indication that there's a cognitive impairment from this very low level exposure to this pesticide, diazinon, and effects on cognitive function. And this is when these animals were in young adulthood. So, and then we can look at the activity, locomotor activity. This is a figure 8 maze. It's the main body of it's in the shape of an 8. There are these photo beams that go across the alleyways to detect the motility of the animal, or how much it locomotes. And you can see over the course of a 1 hour session, they decrease their activity from being considerable early in the session to much less in the later part of the session. And you can see in these stars where there's hyperactivity or hyperlocomotion with the diazinon exposed animals. Actually more in the females than the males. And then you can see on this that the females are more active than the males. And then you have the elevated plus maze, a test of risk-taking versus anxiety. And then in this test, you have the ability of the rat to go to these enclosed arms if they're afraid, or open arms if they're taking a risk. So that's a free choice. And here you can look at the ratio of the time they spend in the open versus closed arms. And you can see, this is the males and females, that there is a significant increase in the percent open arm time. So you have time that's spent in the open times, increased risk-taking with the diazinon exposure. Now, these doses are really quite low, and they don't cause any perceived impact on the health of the animal or the growth of the animal. But in terms of these neurobehavioral effects, this is a very persistent effect. So that this prenatal low-dose diazinon exposure causes long-lasting syndrome of behavioral impairment, including cognitive impairment, locomotor hyperactivity, and increased risk-taking. Very resemblant of a phenotype of what we call attention deficit hyperactivity disorder. So we're continuing to investigate this. We've looked at other pesticides. I'll show you some of those, as well as some heavy metals and organic pollutants. But the next stage of the talk, I want to talk to you a little bit about sex differences in effect and effects on sex differences. And those are two different things. There are males and there are females, and there are a variety of different differences between them. There's lots and lots of overlap, but like height, in general, males are higher stature than females. There's a lot of overlap. But this is also true in other sorts of neurobehavioral functions. Now, in people, it's hard to distinguish between this kind of social impacts of the rearing of boys and girls and how they're brought up and educated and their functions later in life. But in rats, we can more clearly see this, absent the kind of social impacts. So this is a radial arm maze. This is a pretty simple device with, you put the animal in the center, and then there are all these arms radiating out from there. And at the ends of some of the arms are a food bait. They have sweetened cereals that animals like to eat, and they'll go and retrieve these baits and eat them. And then once they've gone to that arm, that's not rebaited, so any effort expended to go back to there is wasted. And like most rational creatures, rats like to optimize their strategy and to get the most reward for the least effort. Some of the arms are never baited, and those are reference memory arms, whereas the arms that are initially baited but not baited thereafter are working memory arms. But here we can see the clear sex differences that are evident in this spatial task. Now, in non-spatial tasks, females actually outperform males, rats. But in these spatial tasks, the opposite is true. So typically there are fewer errors in males, the blue bars, versus the females within the control group. But here, chlorpyrifos, which is another organophosphate pesticide, has a reversal of that effect, so that there is actually the males have more errors than the females, and there's a significant increase in errors in the males and a significant decrease in the females. So this we've seen in a variety of different pesticide exposures, not in chlorpyrifos, but also we see it in parathion, yet another organophosphate pesticide. Here again, we see the increase in errors that is typically seen with the males versus females in the control group with no parathion exposure. But you can see there is a significant increase in errors in the males, but not in the females. In this case, females are better than the control females, but they're not different than the control. So basically, there's a loss of the typical sex difference. And it's not just in terms of cognitive function either. That you see this. This is pre-pulse inhibition. It's a measure of sensory motor response and the plasticity of that. If you have a warning stimulus before a startling stimulus, there's a inhibition of the startle. And here you see this typical sex difference with the zero exposed, the controls, that's wiped out in terms of the exposure to diazinon. And this is postnatal diazinon exposure. But these animals are tested in adulthood, so this is a very long-term effect. So you can see attenuation or even reversal of the typical sex differences with this sort of exposure to pesticides. And we've seen this also with other sort of exposures like marine toxins, like domoic acid, and somewhat in terms of heavy metal exposure as well. So now with the next segment of the talk, I'm going to talk a bit about zebrafish. So they're a very, very nice complementary model to developmental neurotoxicity. And I say complementary rather than alternative because I view that there's a tremendous amount we still don't know about how biology works and how it's impacted by environmental pollutants or other sort of toxicants. So, I've been around long enough to kind of understand that we need to be humble. That each generation typically thinks, yeah, the generations before were relatively uninformed and stupid, and now we know what's going on. And each generation after that says the same. So we have to recognize that there's a lot that we don't know, even though we've learned an awful lot over the course of hundreds of years of research, but there's still a whole lot more to learn. And so that we need these complementary models because there are so many toxicants to be evaluated, and not just the toxicants themselves, but their interactions with each other and their interactions with different subpopulations, males versus females, young versus old, people with different genetic predispositions, people with other sorts of disease states or conditions that make them more vulnerable. And so we need to have higher throughput systems to understand the basic biology of it in terms of effects on cells, in terms of in vitro systems, cell culture systems, invertebrate systems such as fruit flies, Drosophila, or C. elegans, the worms, or aquatic vertebrate systems like zebrafish to complement the rodent models, the non-human primate models, and the clinical and epidemiological testing with people. So here is just a look at zebrafish. So this is, they're just a couple centimeters long. They're native to the South Asia, the areas of India and adjacent countries. And they're very widely used in especially developmental research, because you can see them develop. They have a chorion or egg sac. So they are anatomically and temporally intact system, so that they have that over the in vitro systems, where here, I mean, there's just enormous amount of information that's encapsulated in our evolutionary history, and you can't really tap all of that in terms of looking at cells. Certainly there's evolution of cells and microbes, in particular, but those cells making up an organism is a whole nother leap in terms of our evolutionary history. And so that you need an intact organism for. And they have great tools for visualization of the development and study molecular systems and how they control the development. And they have predictive functional behavioral assessments. And that's our part of it, is to look at these developmental exposures and the effects on behavioral function. So this is some diagrams from my colleague Elwood Linney, and just showing you the fairly rapid development of zebrafish from a zygote to the proliferative phase in terms of organogenesis, somatization, and the development of 3 days until they hatch. And they, have eyes and a mouth and a digestive system and a spinal cord and all the wherewithal it takes to be a freely moving animal. And then you have these transgenic lines where you can light up different parts of the body, including the nervous system, to watch it development. And here's just a diagram of the zebrafish brain from the Woollyman Atlas. And the big part of their brain is the optic tectum. Ours is the cerebral cortex, but this plays a fairly similar role in terms of the processing of where you are, where you need to go, how do you avoid predation, how do you get food. And so we can study the effects in the zebrafish. Here's just an adult zebrafish. It's not related to the zebra, other than they're both winged vertebrates, but has these stripes, and hence the name zebrafish. So you can see they have big eyes, and they locomote about their aquatic environment quite well. And even though the brain is fairly tiny compared to our brain or the brain of a rat, it actually has very similar synaptic and circuit sort of arrangements that the mammalian brains have. So they are fairly predictive in terms of neurobehavioral toxicity. So what we do is we assess them early in life. Here we have typically early life exposure over the first 5 days after fertilization, and then on day 6 we test them for locomotor activity. These are 144 hours post-fertilization, and look at the larval activity. And then we have a series of tests in adulthood, and actually now we're testing the intermediate periods as well in terms of tests of sensory motor function, cognitive function, social interactions, and fear responses. So the larval behavior we test in the 96-well plate, and we can look at their activity whenever it's light They locomote just a little bit. Whenever it's dark, they locomote quite a bit. And this is kind of a evolved predatory avoidance strategy where it's light, they're more vulnerable, and they need to freeze in place to not be preyed upon. Whenever it's dark, they need to forage. So don't be eaten, eat. So whenever it's dark, then they can forage for food. So we can measure that and look at the effects of exposures in a fairly rapid sort of turnover. And here you can see the light and dark changes. And then we have the novel tank diving test, and here we have a fish that, this is a video track where you have, you put them in a novel tank and they tend to dwell at the bottom to begin with as a form of predatory avoidance, and then as time goes on and they're not preyed upon, then they journey to other areas of the water column in a foraging pattern. Tap startle test, we have fish in a little dishes, and you can have these solenoids below to give them a tap, and they would startle and then swim more vigorously for the time after the tap, and then they habituate over the repeated tap. So you can look at plasticity of this response, and then you can look at sociability. So here you have a fish diagram in a tank, and one side has a video of schooling fish or shoaling fish. So there are different types of fish. Some are solitary, some swim in schools, they swim all together as in unison, and some shoal, they swim in and out of each other in a kind of a complex interacting pattern. This is what the zebrafish do. So they're attracted to these shoals. This is a measure of their sociability. And here are the effects of flame retardants. So this is another sort of class of compounds that causes, it inhibits the flammability of clothing or of furniture or other electronic devices. They're pretty widely used for this fashion, and they are actually used in foams that are used in extinguishing fires. And certainly we need to, address the risks associated with fires, but at what cost? We need, like with a therapeutic drug, we need to balance the benefits from the risks. So here we've looked at, this is a mixture called Fire Master 550, and these are the major constituents of them. And then we looked at the effects of exposure to fairly low levels of this Fire Master 550 on sociability in the shoaling test. And you can see here, this is the baseline performance. This is the distance on the left, distance from the image of the of the screen, and this is where without the shoaling. And then you can see on the right-hand side, when there's an active shoal on the screen, the control fish that are only exposed to the solvent DMSO are immediately attracted to that shoal. Whereas these fish who are exposed to low levels of this Firemaster 550 during early development do not show nearly that response. They show a little bit of attraction, but significantly less so than the fish in the active shoal. So then it's a question of, well, can we find flame retardants that don't have this effect? So this is the kind of the next generation, these diphenyl phosphate flame retardants, two of the ones that we investigated. And so they also retard flames, but do they have this sort of neurobehavioral toxicity? So are they suitable alternative to the brominated flame retardants? And here we saw that one of them did not. I mean, it actually also had, this is the distance to the shoal, a significant decrease in the shoaling response. And again, at a very low level. But the other one seemed to be fairly innocuous in terms of its effect on this, even at quite substantial dose levels. So here we can use this sort of tests of neurobehavioral toxicity to get some idea as to compounds that are more risky or less risky. And then I wanted to get into the next phase of the talk on complex mechanisms of neurotoxicity, especially in terms of development. So it's not just a simple relationship of cause and effect, but because organisms are a complex system of interaction, and particularly the brain. The brain's an organ of communication, and so every cell is talking to a variety of other cells, and it's the group actions that are causing the behavior or the dysfunctional behavior. They're incipient mechanisms. So whenever people talk about, oh, here's a mechanism of a certain chemical, and usually they're talking about the incipient mechanism of the receptor that it's acting on, either a neurotransmitter receptor or other sorts of receptor, or an enzyme system. But that's just the beginning of the sequence of actions, and they're followed by integrative mechanisms and also functional mechanisms. So these it's a complex system, and it's not just a passive recipient of insults or stimuli, but it actually reacts to those. So it's not like, one thing causes another, causes another, causes another in a passive fashion, like water going down a series of rapids, but that there are actions and then there are reactions. So that's why sometimes you can have a short-term effect of a low dose of a chemical not be that noticeable because the system's reacting to it. But then over a longer period of time, it could be either those compensatory systems become less able to counteract the effect, or that sometimes those compensatory systems themselves cause adverse effects. And so here we need to kind of take some models to try to understand the complexities of biology. And so that the initial foray into this is what we call the adverse outcome pathway analysis. So you can have this lines of toxicity, this early effect, this initiating effect, molecular initiating effect, and then you have this key intermediate effects, 1, 2, 3, and the other adverse outcomes. In different systems like estrogen signaling or thyroid signaling. Granted, this is also a gross oversimplification. So there's not points of exposure. The zero dimension is not really approached. This is a one-dimensional linear approach. This is an improvement, but it still doesn't encompass what's actually going on. There's an n-dimensional space in terms of lots of different interactions, actions, and reactions of the integrated organism back in compensating for the insult or dealing with it maybe in a not so compensatory way. So anyway, so there are a variety of different adverse outcome pathways that are being developed in terms of trying to describe effects on biology, estrogenic effects, thyroid effects, effects on acetylcholinesterase, the enzyme that breaks down acetylcholine, one of the primary neurotransmitters, effects on vitamin D, or retinoic acid actions. So I just wanted to quickly go through the retinoic acid actions that are related to autism. Patricia Rodier and her colleagues at the University of Rochester came up with this idea 20 to 30 years ago. But basically the brain is like a tube. And so there's this neural plate, there you start, and then it becomes a neural groove, and then a neural tube. And you can see that as this tube closes during development, you get the more adult sort of nervous system. If there's a defect in the anterior or the rostral part, there's you can get microcephaly, very severe sort of brain damage. At the caudal end or the tail end, the spina bifida, very debilitating sort of birth defect. And here you can see the neural plate turning into the neural groove. There's an electron micrograph and the neural tube. And you can get a nice closed neural tube and for further development, but you can get you can get a complete closure, but an inappropriate closure. So as these systems in the brainstem then project to the front part of the brain, the telencephalon, you can get a development, a maldevelopment that would go on to be expressed as autism. So you can perturb the system by valproic acid, which is a drug that is anticonvulsant or anti-epileptic that a number of women would take for treating their epilepsy during pregnancy, and a number of them have children who develop an increased risk for autism. Or excess vitamin A. Here's an essential vitamin, but if you have too much of it, it can cause microcephaly and unlikely autism as well. So we looked at this in fish in the shoaling response. Early developmental exposure to valproic acid caused the decreased shoaling response, as we saw with some of the flame retardants with this valproic acid, and also with excess vitamin A. This, again, is not a very large excess. These animals, to look at them, seem to be quite fine, but they did not show this shoaling response. And then we looked at some pesticides that had effects on retinoic acid receptors, and which of these might be more or less risky. So we found one of them, bupropion exposure, again to very low levels, showed this is measuring the attraction to the shoal. So the number of centimeters from where they were to begin with to where they ended up in terms of the screen. So then we had a significant decrease with this low-level exposure to bupropion. The other ones didn't seem to show this effect. So again, we can distinguish which ones were more or less risky. So that's with synthetic chemicals, but neurotoxic risks are not only from synthesized chemicals, also from natural elements. I mean, you say, oh, well, lead's been around for our entire evolutionary history, should be fairly innocuous. But the fact is that it's not. The environmental distribution of lead has been really greatly altered by human activity. And here, this is just showing over the last couple hundred years, the lead levels. The lead that's always been there has always been largely locked up and away from our bodies. But as we went through the Industrial Revolution and started mining this in a big way, then it got more and more in our biosphere and in our bodies, so that it became a big problem. And it has more pronounced effects during development compared to adults. And this has been known for a while. Here's a paper from 1932 showing lead poisoning in children. And here we've had lead in gasoline, lead in paint, lead in plumbing. Actually, the word plumbing comes from the Latin word for lead, because early plumbing was made of lead because it's quite malleable. And even more recent plumbing in terms of lead in the joints between the tubes, because it's used in solder. And we've had terrible problems in the U.S. and in other parts of the world where you have, particularly in older housing, where the pipes have not been changed, that you would have lead in the drinking water. And it has been shown that developmental lead exposure does lower IQ. And this is just I'm looking at several different studies, and it's a small effect, somewhere between 5 and 10% a decrease in IQ. This is combined showing just a couple points lower, and then in combination with other things, maybe a couple points more. So it's not a huge effect, but it's effect across entire populations. And this is again showing you the cumulative distribution. And not only you see effects decreasing it, but also fewer and fewer of the higher IQ individuals. And so not only is it in terms of cognitive function, but other problems are seen as well. And this is in terms of aggression and delinquency in different studies, the high lead versus the low lead groups, so that you see effects in terms of this emotional control as well. So we've looked at animal models to determine the mechanisms of toxicity and develop therapeutic treatments. And so here we have, we did some work with rhesus monkeys. This is back in decades ago when I was a graduate student. These are rhesus monkeys exposed to low levels of lead, didn't impair their physical health or well-being, but did cause a persisting impairment in their choice behavior. And so, if I go back a slide, well, actually not. You have this lower percent correct in terms of choosing which stimulus had the food reward below it. And here you see over delays of 5, 10, 20, 40 seconds, the controls did quite nicely, high percent correct early on. As time delays increased, they did not so well. Here from the very beginning, that there was an impairment with the lead-treated monkeys. And this is, again, when they're adults, so very long-term effect. So it seemed to be an intentional effect. And then we tried a therapy, L-DOPA, which is a therapy to help people with Parkinson's disease to increase their dopamine. Lead is shown to actually impair dopamine systems. And here we saw with several weeks of exposure to L-DOPA, given in pitted prunes, actually, that there was a gradual improvement in their choice accuracy up to be just slightly lower, but not significantly lower than controls. And actually, in the later part of the study, we took it away and that resumed the impairment. So here we saw something that would be therapeutic for the longer-term effect. And so it's important to develop these therapeutic treatments because, ideally, you don't get exposed in the first place, and you have this sort of, avoiding the problem. But then there's ourselves, me included. I grew up in a polluted environment. How do you treat the problem that's already there? And these people who are really have to seek them out so much as they're already there. They're in our clinics and already. It's just that some people may have a certain impairment because of genetics or disease or environmental exposure. So how are they differentially helped by a therapeutic treatment that addresses the systems that are affected by the toxicants they've been exposed to? So we have also looked at this in rats. This is a much simpler 8-arm maze, similar to the 16-arm maze we did earlier. And this is the number of entries that they had to repeat, and you see in errors that are increased with the lead exposure groups. And here you saw, this is pergolide, which is a dopamine agonist. So this is a drug that stimulates dopamine receptors, much in the way L-DOPA does indirectly. And you see that this is improves actually both the controls and the lead-treated rats, but to the point where there's no apparent impairment compared to the control baseline performance. And then there's another sort of treatment, pilocarpine, which is an agonist or stimulating the receptor, acetylcholine receptors, muscarinic acetylcholine receptors. And again, here's the impairment seen without the treatment. And then here you see the improvement with this treatment with the lead-treated rats. So developmental lead exposure lowers IQ, increases aggression. Recent safety threshold of 10 micrograms per deciliter in the blood really gives you no threshold, safety threshold, no margin for differential sensitivity. Certainly lower levels have been shown to have effects. Therapeutic treatments may be possible, as we've seen with the dopamine and acetylcholine treatments. The best option is to reduce the exposure, but we do need to develop treatments as well. So, in conclusion, neurotoxicology is important. I hope I've convinced you of that. Adverse effects are seen even at quite low levels of exposure, and some of these effects are subtle, but I would convey to you the idea that there's this pyramidal shaped dose-effect function, where at the peak levels of exposure, there are relatively few people exposed with a very dire effects, but as you go down in dose, there's fewer and fewer effects, more subtle effects, but a broader proportion of the population are affected. And so that we need to be on guard to as well, and I'll speak to that in a minute. Then we have effects on sex differences. And these are, naturally occurring phenomena which, are certainly need to be attended to as well. And the zebrafish and other sort of complementary models offer valuable models that would complement the typical rodent and other mammalian models. Neurotoxicity is also seen with natural chemicals, like elements like lead, or other sorts of natural plant materials or animal materials. And therapeutic treatments can be used to counteract the effects. Now, I just want to go into this population changes just for the last little bit here. And here, Bernie Weiss from the University of Rochester came up with this originally. This is his original diagram, but this is a bell-shaped curve of the normal distribution of IQ scores centering on 100. And this is what would happen if you would go down to 95, a decrease, that there would be both an increase in people who have below 70 or impairing cognitive impairments in their IQ, and then a decrease in this upper limb of, in this room, IQs above 130. This actually would be a little easier to see, and Charles Schmidt actually looked at the same. It's a little easier to see in his graph back in 2013 in Environmental Health Perspectives, this change, same idea of increased numbers of people with the impairments and cognitive function decreased in the gifted, you would say. So in a population, you might recognize this figure, 85.82 million people in Iran. Decrease of 5 IQ points. Well, if you have a mean of 100, then below 70 it would be almost 2 million people. Above 130, almost 2 million people. Mean of 95. Here's 3.09 million. Now this is increase of some 50, more than 50%. But here a decrease of over half of the people in the gifted category, 0.79. So you have a greater number of people who need care, and fewer number of people to come up with new understanding and therapeutics. So there you are. So here is the people have done these calculations, Grandjean and Landrigan and Lancet looked at the IQ points lost, come up with these very large numbers, preterm birth, autism, various pediatric and adolescent disorders, and then lead, substantial. Mercury maybe less so because it's less widespread. Organophosphate pesticides, also a big problem, and other toxicants. And Tresand and Leo came up with this costs of, if you want to bring it down to dollars and cents, $50 billion in the U.S. From the annual cost of lead poisoning, and $5 billion to methylmercury. And what are the reasons for addressing neurotoxicity? I would put out to you that the primary reason is ethical, that it would be the right thing to do to have people not suffer impairments for the rest of their life from early life exposures. That's just an ethical construct, right? There's a medical issue. People are sick, and we are here to help keep people from being sick and to make them better. And if this is what it takes, there's the economic. So, there are a number of reasons. If it takes going down to the economic, so be it. There is an economic imperative. I mean, it's costing society a lot in terms of ignoring these things. So you say, oh, people would lose their jobs if we abandon this or that industry. It's like, what's the cost of doing nothing? Times it can be considerable. So progress should be, measured on a variety of different ways. Not doing ill to our fellow people is a big one. And finally, I just want to leave you with this map of North America in 1650. So bring you back to the side of the world where I live. This is 158 years after Columbus came over to this area here. I mean, it's not to mention that tens of thousands of years before that, people came over in the other directions and not only discovered but completely settled North and South America. But in terms of the Europeans coming by, Columbus came here. It took 150 years to come up with this map. Some of it they had fairly accurately in terms of the East Coast and the Caribbean. Some of it was terra incognita. They recognized that they didn't know, but we need to recognize there is terra miscognita, like California is not an island. And so even quite some time after we think we know, there are things that we don't know, but we recognize we don't know them. But there are things we think we know, but are really quite wrong. So we have to humble ourselves to understand that there's much that we don't understand, but to not let that keep us from making further progress. And certainly we can make this progress generation after generation, and we need to add to this sum of knowledge and also bring that to bear in terms of better regulation and better treatment of our fellow citizens of the earth. Okay, I wanted to thank you very much for inviting me, and I would be happy to respond to any of your questions. If I'm not available in a live fashion, feel free to email me any questions, edlevin@duke.edu. So edlevin@duke.edu. And I thank you so much for your attention.
Thank you, Professor Levin. That was very good presentation you had. I think that Professor Levin is connected. If he's connected, if any of the any question, I think he could respond to that. Anybody have any question? Well, I thought he's connected. He's actually disconnected just right now. خب خیلی ممنون از همه کسانی که تا این وقت در کنار ما بودن. انشالله که در واقع استفاده های گوناگون علمی همه همکاران برده باشن. ما فردا صبح از ساعت 8 با اورال پرزنتیشن شروع میکنیم و فکر میکنم بعد از ظهر هستش که سمپوزیوم های مختلف و یک کینوت اسپیکر هم دوباره در انتهای روز هستش. ما برای اینکه همکار کسانی که الان دارن صدای من دارن در جریان باشن، ما به خاطر اینکه تعدادی از این اساتید میخواستن آنلاین در واقع پرزنتیشن داشته باشن، ما ساعت کینوت اسپیکر ها و در واقع سخنرانی‌های خارج از کشور رو در زمانی گذاشتیم که تا یه حدودی اون زمانی باشه که زمان در واقع نصف شب نباشه و برای همین ما اینا رو بعد از ظهر گذاشتیم. ولی خب متأسفانه یه تعدادی از حضار رو اینجوری داریم از دست میدیم، ولی به هر حال سخنرانی خیلی خوبی هستش که پیشنهاد میکنم که همگی تا یه حدود زیادی، مخصوصا کینوت اسپیکرها که واقعا زحمت زیادی کشیده شده که تا اینها سخنرانی خودشون آماده کنن، بتونن هم دانشیان، اساتید و همکاران گرامی من بتونن استفاده ببرن. انشالله فردا صبح ساعت 8 روز دوم کنگره رو شروع میکنه. خدا حافظ.